In this session, we are going to start one of the most important scoring units for GAT-B, that is biomolecules. This topic forms the base for many advanced topics like metabolism, enzymes, and bioenergetics. So, it is a very important that you understand each concept clearly rather than just memorizing. We will cover all major areas step by step in a logical sequence so that you can connect concepts easily. While studying, always try to relate structure with function because most exam questions are based on application. If your fundamentals are strong, you can solve even tricky MCQs confidently. As you can see in this slide, our focus is on concept-based learning. That means we are not just studying theory, but understanding the logic behind each topic and concepts. This approach helps you to retain information for a longer time and apply it in exams. We also focuses on important MCQs, previous year questions, and a structured preparation strategy. Many students study randomly, but here the preparation is systematic, from understanding concepts to solving questions, and finally exam guidance. This ensures that you are fully prepared for GAT-B. In this unit, we are going to study multiple interconnected topics. We will begin with structure and function of biomolecules, then move towards membranes and transport, followed by enzymes, metabolism, and finally bioenergetics. It is important to understand that all these topics are linked. For example, enzymes control metabolic pathways, membranes regulate transport, and bioenergetics explains how energy flows in these processes. So always try to study these topics in an integrated way rather than separately. Introduction to biomolecules. Biomolecules are chemical compounds present in living organisms, and they are essential for life. They help in maintaining structure, carrying out functions, and regulating cellular processes. Without biomolecules, no biological activity can take place. There are four major classes of biomolecules: carbohydrates, proteins, lipids, and nucleic acids. Each has a specific role in the cell. For example, carbohydrates mainly provide energy, proteins perform functional roles, lipids are involved in membrane structure, and nucleic acids store genetic information. Carbohydrates structure. Carbohydrates generally follow the formula CH2ON and are one of the primary biomolecules in living systems. They are classified based on the number of sugar units present. Monosaccharides like glucose are the simplest form. Disaccharides like sucrose consist of 2 units, and polysaccharides like starch and glycogen are complex molecules. These sugar units are linked by glycosidic bonds. Understanding classification and bond types is very important for exam. Carbohydrates function. Carbohydrates mainly act as an energy source for cells. Glucose is the most important molecule that is broken down to produce ATP, which is the energy currency of the cell. This makes carbohydrates essential for survival. Apart from energy, carbohydrates also perform structural roles. For example, cellulose forms the plant cell wall. They are also involved in cell recognition through glycoproteins and glycolipids. So, carbohydrates have both functional and structural importance. Proteins structure. Proteins are made up of amino acids, and there are 20 standard amino acids that combine in different sequences to form proteins. The sequence of amino acids determines the final structure and function of the protein. There are four levels of protein structure: primary, secondary, tertiary, and quaternary. Primary is the amino acid sequence. Secondary includes structures like alpha helix and beta sheet. Tertiary is the 3D folding, and quaternary involves multiple subunits. Questions often focus on these levels, so understand them clearly. Proteins function. Proteins perform a wide variety of functions in the body. One of the most important roles is enzymatic activity, where proteins act as enzymes to speed up biochemical reactions. Apart from that, proteins provide structural support, help in transport of molecules like oxygen, act as hormones and receptors, also play a role in immunity through antibodies. Because of this diversity, proteins are considered the most versatile biomolecules. Lipids, structure and types. Lipids are hydrophobic molecules, which means they do not dissolve in water. This property makes them very important for forming biological membranes. There are different types of lipids, such as fatty acids, triglycerides, phospholipids, and steroids. Phospholipids are especially important because they form the bilayer of cell membranes. Steroids like cholesterol help in maintaining membrane stability and also act as hormones. Lipids function. Lipids are mainly used for energy storage and they provide more energy compared to carbohydrates. This makes them efficient, longer time energy storage molecules. There are also plays an important role in forming cell membranes, act as hormones in the case of steroids, and provide insulation and protection to the body. So, lipids are not just fats, they are essential for maintaining cellular structure and function. Nucleic acids. In this slide, we are focusing on nucleic acids, which include DNA and RNA. These are extremely important biomolecules because they are responsible for storage and transmitting genetic information. Without nucleic acids, heredity and protein synthesis cannot occur. Each nucleic acid is made up of basic units called nucleotides. A nucleotide consists of three components: a sugar, a phosphate group, and a nitrogenous base. DNA mainly stores genetic information, while RNA plays a crucial role in protein synthesis. Questions are often asked on structure and difference between DNA and RNA, so be clear on this. Now we are moving into a new section, which is biological membranes. This is a very important topic because membranes are essential for maintaining the integrity of the cell and controlling what enters and leaves the cell. Biological membranes are not just barriers, there are dynamic structures involved in communication, transport, and signaling. Understanding membrane structure and function is important because many physiological processes depend on it, and questions are frequently asked from this area. Membrane structure. As shown in this slide, the membrane follows the fluid mosaic model. This means the membrane is fluid in nature and contains a mosaic of different components like lipids and proteins. The main structure is a phospholipid bilayer, where phospholipids are arranged with hydrophilic heads outwards and hydrophobic tails inwards. Proteins can be integral or peripheral, and cholesterol helps in maintaining fluidity. The phospholipids is the key concept here, and it is very important for exam. Membrane functions. The cell membrane performs several important functions. One of the key functions is selective permeability, which means it allows some substance to pass while restricting others. It is also involved in cell communication through receptors and signaling molecules. Additionally, it helps in the transport of molecules and maintains internal balance, which is called homeostasis. So, the membrane is not just a boundary, it is an active and functional structure. Transport processes. Transport across the membrane can occur through different mechanisms. Passive transport includes diffusion, facilitated diffusion, and osmosis, where no energy is required and movement occurs along the concentration gradient. Active transport, on the other hand, requires energy in the form of ATP, like in the sodium-potassium pump. There is also vesicular transport, which includes endocytosis and exocytosis. Understanding difference between these transport types is very important for MCQs. Action potential is an electrical signal generated in neurons, and it is essential for nerve impulse transmission. It involves changes in membrane potential due to movement of ions. Initially, the cell is at resting potential. During depolarization, sodium ions enters the cell, making the inside more positive. The repolarization occurs when potassium ions leave the cell. This sequence allows the signal to travel along the neuron. Questions often test the sequence and ion movements. Now we are starting the enzyme session, which is one of the most important and frequently asked topics in GAT-B. Enzymes are biological catalysts that speed up chemical reactions without being consumed. They are highly specific and act on particular substrates. Enzymes lower the activation energy of reactions, making processes faster and more efficient. Understanding enzymes' properties is essential because many questions are concept based. Enzyme basics. Enzymes function by lowering the activation energy required for a reaction to occur. This allows biochemical reactions to happen quickly under normal physiological conditions. Another important feature is specificity. Each enzyme works on a specific substrate, forming an enzyme substrate complex. Because of this specificity, even small changes in structure can affect enzyme function. This concept is very important for understanding inhibition and mechanisms. Enzymes are classified into six major classes based on the type of reaction they catalyze. These include oxidoreductase, transferase, hydrolysis, lysase, isomerase, and ligases. Each class has a specific role. For example, oxidoreductase are involved in redox reactions, while hydrolase breaks bond using water. Questions are often direct from classifications, so try to remember at least one example for each class. Enzyme kinetics. Enzyme kinetics deals with the rate of enzyme catalyzed reaction. The equation is used to describe this relationship between substrate concentration and reaction rate. Two important terms are Vmax, which is the maximum reaction velocity, and Km, which indicates substrate affinity. A low Km means high affinity. The graph is typically hyperbolic. This topic is very important and often asked in numerical or conceptual questions. Enzyme inhibition. In this slide, we are discussing enzyme inhibition, which is very important from an exam point of view. Enzyme inhibitors are molecules that reduce or stop enzyme activity. This concept is widely used in drug design and regulation of metabolic pathways. There are different types of inhibition. In competitive inhibition, the inhibitor binds to the active site and competes with the substrate. In non-competitive inhibition, the inhibitor binds to a different site, affecting enzyme function without blocking the active site. Uncompetitive inhibition binds only to the enzyme substrate complex. Understanding differences between these is very important for MCQs. Now, let us understand how enzymes actually work. There are two main models, the lock and key model and the induced fit model. The lock and key model suggests that the enzyme and substrate fit perfectly like a key into a lock. However, the induced fit model is more accepted where the enzyme changes its shape slightly to fit the substrate. This helps in stabilizing the transition state and lowering activation energy. This concept of transmission state stabilization is very important as it is explained how enzymes increase reaction rates. Now we are moving into metabolism, which is the sum of all chemical reactions occurring in the body. These reactions are essential for maintaining life and supporting cellular functions. Metabolism is broadly divided into two types, catabolism and anabolism. Catabolism involves breakdown of complex molecules to release energy, while anabolism involves synthesis of complex molecules using energy. Understanding these differences is very important because many questions test these basic techniques. Overview of metabolism. As shown in the slide, metabolism includes all biochemical reactions taking place inside a cell. These reactions are highly regulated and interconnected to maintain proper functioning of the organism. Catabolic pathways generate energy by breaking down molecules, where anabolic pathways use the energy to build up complex molecules. The balance between these two processes is very important for maintaining homeostasis. Any imbalance can lead to metabolic disorders. Carbohydrate metabolism includes several important pathways. The first step is glycolysis, where glucose is broken down into pyruvate, producing a small amount of ATP. After glycolysis, pyruvate enters the Krebs cycle, which generates high energy molecules like NADH and FADH2. There are also other processes like gluconeogenesis, where glucose is synthesized, and glycogen metabolism, which involves storage and breakdown of glycogen. These pathways are very important for exams. Lipid metabolism mainly involves the breakdown and synthesis of fats. The breakdown of fatty acids occurs through a process called beta oxidation, which generates acetyl-CoA. Lipogenesis is the process of synthesizing fatty acids, while ketone bodies are formed during prolonged fasting or low carbohydrate conditions. These concepts are important because they connect metabolism with physiology conditions like starvation. Amino acid metabolism involves processes like transamination and deamination. In transamination, amino groups are transferred between molecules, while in deamination, the amino group is removed. The ammonia produced is toxic, so it converted into urea through the urea cycle. This is very important for Detoxification. Questions often focus on the processes and their significance in nitrogen metabolism. Nucleotide metabolism involves the synthesis and breakdown of purines and pyrimidines. These are essential for DNA and RNA formation. There are also salvage pathways, where the cell reuses bases instead of synthesizing them again. This is energy efficient. Understanding these pathways is important because defects can lead to diseases, and such concepts are sometimes asked in exams. Now we are moving to photosynthesis, which is the process by plants convert light energy into chemical energy. This process is essential for life on Earth as it produces oxygen and food. Photosynthesis occurs in chloroplast and involves conversion of carbon dioxide and water into glucose and oxygen. This process forms the base of the food chain, and understanding its steps is important for conceptual clarity. Photosynthesis involves conversion of light energy into chemical energy in the form of glucose. The overall equation shows carbon dioxide and water forming glucose and oxygen. This process is divided into different stages, which we will study in the next slide. Understanding the overall concept first is important before going into detailed steps. Questions are often based on the equation and location of the processes. In this slide, we are focusing on the light reaction of photosynthesis. These reactions take place in the thylakoid membrane of chloroplast and require light energy. The main purpose of this stage is to convert light energy into chemical energy. During light reactions, ATP and NADPH are produced, which are later used in the Calvin cycle. Two important components involved are photosystem 1 and photosystem 2. These systems absorb light and help in electron transfer. Understanding the sequence of electron flow and role of these photosystems is very important for exams. Now we move to the dark reactions, also known as the Calvin cycle. These reactions occurred in the stroma of the chloroplast and do not directly require light, but they depend on ATP and NADPH produced in the light reactions. In this cycle, carbon dioxide is fixed and converted into glucose. The key enzyme involved is Rubisco, which is one of the most abundant enzymes on Earth. Questions are often asked about CO2 fixation and the role of Rubisco, so this concept should be very clear. Now we are entering another important topic, cellular respiration and the electron transport chain. This process is basically the opposite of photosynthesis, where glucose is broken down to release energy. Respiration involves multiple steps and ultimately leads to the ATP production. The electron transport chain plays a major role in generating maximum ATP. Understanding how energy is released and captured during respiration is very important for conceptual clarity. Cellular respiration is an anaerobic process, meaning it requires oxygen. It involves three major steps: glycolysis, Krebs cycle, and the electron transport chain. In glycolysis, glucose is broken down into pyruvate. The Krebs cycle further processes these molecules to generate electron carriers like NADH and FADH2. These carriers then enter the electron transport chain. Understanding the sequence of these steps is very important for exams. The electron transport chain is located in the inner mitochondrial membrane. It involves a series of protein complexes that transfer electrons from NADH and FADH2. As electrons move through the chain, a proton gradient is created across the membrane. This gradient is suddenly or used to create or produce ATP. This concept is very important because it explains how most ATP is generated in the cell. Oxidative phosphorylation is the final step of cellular respiration where ATP is produced using the proton gradient generated in the electron transport chain. ATP synthase is the key enzyme that uses this gradient to synthesize ATP. This process is explained by the chemiosmotic theory. Since this step produces the maximum ATP, it is highly important from an exam point of view. Now we move into bioenergetics, which is the study of energy flow in biological systems. This topic connects all the previous concepts like metabolism, respiration, and photosynthesis. Bioenergetics helps us understand how energy is produced, stored, and utilized in living organisms. ATP plays a central role in the process and is known as the energy currency of the cell. Bioenergetics basics. As shown in this slide, bioenergetics mainly deals with how energy flows in cells. Every cellular process requires energy, whether it is transport, synthesis, or movement. ATP is the main molecule that stores and transfers energy, where ATP is broken down into ADP and inorganic phosphate. Energy is released. This energy is then used to perform various cellular activities. Thermodynamics explains the principles of energy transformation in biological systems. The first law states the energy cannot be created or destroyed, only converted from one form to another. The second law states that entropy or disorder always increases. In biological systems, this means energy transformations are not 100% efficient. Free energy change, represented by delta G, determines whether a reaction is spontaneous. Negative delta G indicates a spontaneous process. ATP is the main energy currency of the cell. When ATP is hydrolyzed into ADP and inorganic phosphate, energy is released and used for cellular work. This energy is used to processes like active transport, biosynthesis, and muscle contraction. Understanding how ATP is utilized is very important because almost every biological processes depend on it. In this slide, we are discussing high-energy compounds, which plays a crucial role in cellular metabolism. These molecules store and transfer energy required for various biological processes. The most important among them is ATP, but there are other molecules also involved. NADH and FADH2 are electron carriers that store high-energy electrons during metabolic reactions. These electrons are later used in the electron transport chain to generate ATP. Acetyl-CoA is another important molecule that links different metabolic pathways, especially carbohydrate and lipid metabolism. So, these compounds act as energy carriers and connectors in metabolism. Integration of metabolism. Now, let us understand how different metabolic pathways are interconnected in the body. Carbohydrate, lipid, and protein metabolism do not occur separately. They are all linked to each other. For example, excess carbohydrates can be converted into fats and amino acids can enter metabolic pathways at different points. This integration allows the body to adapt to different conditions like fasting or feeding. Regulation of these pathways occurs through which is very important for maintaining balance. We are summarizing all the concepts we have studied. Biomolecules form the structural and functional basis of life, and each class has a specific role in the cell. Enzymes regulate metabolic pathways, and energy is produced through processes like respiration and the electron transport chain. Photosynthesis provides energy input for ecosystems, and bioenergetics explains how energy flow is transferred and utilized. This overall integration is important for understanding life processes. From an exam point of view, you should focus on key areas like enzyme kinetics, including Km and Vmax, and metabolic pathways such as glycolysis and their electron transport chain. Also pay special attention to the membrane transport mechanisms and enzyme inhibition types. Practice diagrams, cycles, and MCQs regularly. Consistent revision is very important because this unit has many interrelated concepts that can be easily forgotten if not revised properly. Finally, stay consistent with your preparation and keep updating yourself with the latest exam patterns and important topics. Regular practice and revision will help you improve your confidence and performance. Subscribe to BioBrainz. Follow us for more GAT-B 2026 updates, preparation tips, and lectures. Remember, simple preparation, strong concepts, better results. Thank you for watching.